Good day, everyone. Today we investigated the fascinating field of medical entomology, and we have an interesting tale to share.
We began our entomology in our first laboratory exercise by knowing the internal and external parts of insects and arachnids and their function. We started with the antenna, which was a fascinating feature. We learned that insects have many different forms of antennae, and they also have various functions. We then discovered a world of diversity in the mouthparts. The mouthpieces are evidence of how adaptable and resourceful these amazing creatures are. The legs prove that the amazing range of tasks that insects perform in their habitats is highlighted by the fact that each leg type is designed for a particular function. Finally, we know that the wings of insects occur in a variety of sizes and shapes. Our thorough knowledge of insect anatomy provided a solid foundation for our further forays into collecting and identifying arthropods.
In our second laboratory, we used different collections, the active and passive collection. In our active sampling, we did a general hand collection, and based on our experience, this collection was more accessible since there are already multiple species that we could get by just using our hands. On the other hand, using the collecting tool sweep net allows us to capture more prominent species like those that are flying. Still, from our experience, one should be entirely concentrated when doing this since some species from this class, Arthropoda, flies really fast. Our journey took place at Nicanor Reyes Hall where we encountered a wide range of fascinating arthropods including black flies, centipede, and spiders.
Our journey did not finish there. Though with the help of sneaky concoction of century tuna and peanut butter, we were able to entice the ants. We put a lot of baitings in different parts of the arts building, 5 from the inside and 5 from the outside. After a few minutes, we saw that multiple ants were baited from certain places. However, in some areas, there were no baited ants.
Now discuss passive collection, which is held in arts building. Our baits are consist of peanut butter and tuna mix and enticingly arranged on a 1/4 paper. After making the bait, we put the baitings in different positions and waited for 30 minutes. We used an alcohol to sedate our capture after. We are beginning to recognize the outstanding diversity of our environment around us.
The adventure continued after that for us to identify the type of species we caught. Each ant that we captured was put in a petri plate using forceps that contained 90% ethanol, and it was then moved into another petri plate that contained distilled water. After this, we preserve it in a test tube that contained 90% alcohol, and then we meticulously documented our findings for better identification of ants that we have.
For our third laboratory activity, we studied the craft of mounting insects, while the ants were carefully pinned on paper stages. The ants were pinned through the thorax and positioned with their heads facing the left. Then, using a dissecting microscope, we look at them from a hashed ventral and dorsal view. These observations served as the foundation of our documentation, which was an important step in the identification procedure. Let me further elaborate. Entomological mounting refers to the preparation of an arrangement where pinned insect specimen papers are placed. This step serves to keep specimens in place and provide for identification and archiving. The materials we used for this are the following: entomological puncher, which is used to make the paper stage, sturdy paper, entomological pins, forceps, glue, as well as ruler and pencil. In addition, just a short information about dichotomous key. It is a hierarchical system used to identify organisms in biology and taxonomy. The process involves a set of two-part comparisons which ends with specific categorization. Dichotomous key are versatile. They may be illustrated and can nowadays even be obtained in digital format. They are useful in fieldwork, education, and systematic identification, although they can be of no use for some variable 1. And lastly, the ability to use them takes experience and knowledge of the insect to be recognized.
That concludes our fascinating excursion into the world of the arthropods, discovering the mysteries of these extraordinary creatures and their value in the field of the health is the heart of the medical entomology, which is more than just a collection of insects. We came to understand the insects serve as the foundation for advanced biological and chemical control of insecticides and pesticides, medicine, food production and storage, biological diversity, mapping, and in other scientific domains. Several people might think that these species are just pests. Still, due to the learning of entomology, we realize that these species also provide help for us because without their pollinating services, we would only have a few of the crops we love and rely on. Lastly, these species help us to decompose waste and organic matter, promoting new plant development, and helps to ensure that ecosystem maintain adequate nutrients.